so I hope you guys enjoyed the video. Now we're gonna do a short Q&A session with Nizam. So if you have any questions, you can drop them in the chat box and please raise your hands. So we'll keep watching who's been sending the questions. So Nizam, the first question is from Anushka Sarkar. She's asked, can we use brain on a chip to mimic and study neurodevelopmental disorders, and how can we go about it?
Hi, Anushka. Thank you for the question. First of all, I mean, when we talk about organ on a chip worldwide, it's progressed a lot. So people are also using brain on a chip. Basically, what they do is they would use your basic cells like glial cells and brain cells uh, on a chip, and they would try to study it. Now, our expertise is basically wound infection on a chip, and we focus on that. But if we talk about the progress in the field overall, yes, that is happening, and that is very much possible.
Okay, great. Yugang has asked, are there any other alternatives to trypsinization?
Yeah, so yes, there are alternatives to trypsinization. Now these cells, your keratinocytes and fibroblasts, they are very well stuck. Now keratinocytes in itself, they're compactly packed. So for such cells, we also use something of a physical nature, like it's a brush kind of sterile, I can't call it device, but then it's an apparatus that is used to brush it off, brush the cells on the bottom of the flask, and that is the one alternative I know of trypsinization. It's basically to get rid of all the cells. So that is basically physical aberration. Yeah.
Okay. For the next question, I'm going to ask everyone to unmute themselves so you can ask your questions yourself. I think that'll be more fun. So, Shravani, can you please unmute yourself and turn on your video and ask your question?
Yes. Actually, I wanted to know how we can use organ-on-chip devices for antimicrobial resistance.
Yes, for sure. So you can certainly use. So the main idea is to recapitulate this particular. Now we study antimicrobial resistance on, say, plates, right? But how relevant are they in cases of, say, wound or other sites? So what we can do is we can certainly add the environment. For example, if you talk about wound, we can add skin cells, we can add shear stress, etc., etc. But apart from that, one important component will be your resistant organisms, right? And then you feed, instead of feeding using your normal medium, what you can do is you can flow through antibiotics where you want to test it, and you can always check on fluorescence and see a few methods. So you can do that as well. The main idea is to recapitulate as much as you can, because I mean, we are not plates. Basically, we are much more complex than normal nutrient medium plates.
Okay, Sachin, you can go ahead and ask your questions. Sachin, are you there?
No problem. I can read it.
I think he's unmuted himself. Okay, maybe we'll move on to the next person. We'll come back to you, Sachin. Khushi?
Yes, ma'am. Am I audible?
Yeah, you can go ahead.
Yeah, you are.
So, which
Hi.
what concentration or percentage of trypsin had you used in this, and which cold or warm trypsination which is preferred for this type of cell?
So I use 0.25% of trypsin, which is along with the EDTA, and mostly it is warm trypsinization. Actually, it's under 37 degrees Celsius.
Sir, in the
I hope
case of
that.
warm trypsinization, the efficiency won't decrease of the viable cells.
It does not actually. I mean, because we have checked it by warm trypsinization. I mean, at 37 degrees Celsius. Now, in order to check the efficiency of cells, what we also do is we count it using Trypan blue exclusion assay. And I don't happen to see a lot of dead cells in that. So we do retain a good amount of viable cells.
Okay, sir. Thank you.
Yeah, you're welcome. You're welcome.
Okay, Sachin, you can go ahead.
His mic is not working. I can answer his question.
Oh, okay. So
Yeah.
yeah. So I'll just read out the questions that he asked. How can we implement the understanding of wound healing which occurs in cancer cells using the chip? And can you create a physical wound on a chip like you create on a well plate?
Yeah. Okay. So I think there is one more question on physical wounds, right? To sort of mix both the questions. Now we can study now. Most importantly, what we have, the device that we have, is macrofluidic in nature. So not only can you grow cells in it, you can also study wound migration and proliferation, something called as in vitro scratch assay. Now, what we do is in an in vitro scratch assay, we will, make a scratch using a 200 ml pipette tip, and we will try to study cells, closing, migrating and proliferating to close the gaps. Now, this is how a physical wound is created. Secondly, about your, yes, you can also study cancer cells. So instead of using your normal keratinocytes and fibroblasts, you can use your cancerous cells, to as your cell types, and you can certainly study that as well.
Okay. Next question is Yugang.
Am I audible?
Hi. Yeah.
Go ahead.
Yeah, so I actually had two questions. The first one was that do these organ-on-chip models play a role in modeling organoids that are being used in research, and are there any limitations associated with that?
Whether they are used for organoid development, I don't think. I mean, I'm not very sure of the area of organoid as such. So I would not be the right person to answer this question. I'm sure there will be a lot of follow-up videos on organoid development, because we don't work on modeling as such. So I would be the wrong person to answer this question. I'm so sorry.
Okay.
So
there was
Yugang,
another question.
you should look out for the next video, which is going to be on
Next
organoids.
video,
Okay.
Yeah.
But there was one more question, if I may ask.
Then, yep, sure.
The peristalsis device that was mentioned in order to create the shear stress. So are there similar devices when we are developing organ-on-chip models for different organs, heart, liver? If so, then how are those models made? All right.
Okay. So now what we made, we could see the tubings, right? The diameter of the tubings, they change. Now this peristaltic pump that we developed, it's cheap. It's low cost. It's developed in a lab itself. Now, what other labs also do is they use peristaltic pumps that are bought from the market. They're pretty expensive, but also the diameter sort of changes. So the diameter will also depend on how much crevice will your microfluidic device have. So there are different variations of peristaltic pump that are used. So some people we use a continuous flow, for example. Some people might use a pulsed flow. Like, they would want flow for some time and then not for some time in a cyclic manner. So there are certainly different kinds of peristaltic pump that are used, for sure. Yes, also depending on the diameter of the tubing.
Okay, thank you.
Yugang, just to answer your first one, actually there are labs which are now incorporating both organ-on-chip and organoids. So for example, organ-on-chip is basically a microfluidic device where you can, pass fluids in a very at a very micro level. So there are labs which are incorporating organoids into these kind of microfluidic devices also, which is also a very interesting area. So yeah, so like Anushka said, we'll be covering organoids, but yeah, just to give you a small thing, when you can look up research papers, and maybe we'll also forward some where people are incorporating both these kind of model systems together. Yeah.
Thank you.
Okay, Yogita, you can go ahead and ask your question.
I just want to say a line here that I would love to congratulate Kaushik Lab for developing this peristaltic pump, because we've been trying to analyze how much it costs, and the costs are in lakhs, if nothing.
Yes, it is.
And they have developed it just within thousands. So this is a great move forward, and I really love that they are doing something to do something with indigenous supplies.
Thank you
Okay,
so much,
Yogita, you
actually.
can
It's
go
been-
ahead.
She's saying,
okay,
please answer
just
my
read
question.
it out.
Yeah.
Yeah.
Yeah. So Yogita has asked, can we use microtomy technique instead of a chip to mimic the model of your wound created?
Uh, Again, I have limited knowledge about microtomy technique, actually. I'm so sorry. But you know what? I will also do one thing. I will get back to you all. You can write me on my email, and I can get back to you. I don't have pretty good knowledge about microtomy technique as such. But I will get back to you. You can write to me, and I'll read it up for sure. I will read it up for sure.
Nizam, maybe you can just drop your
I will
Twitter handle
do that.
in the chat box. So if anyone
Actually,
has any questions,
I
you
will
can
do
reach
that.
out to me. Okay. Nanika, you can ask your question.
Hello, good morning.
Hi. Hello, good morning.
Yes, sir. The question is, can trypsin overchop the cell that are attached to the vessel wall?
Okay. Good question. No, well, first of all, trypsin doesn't chop the cells as such. But yes, what happens is when you keep the cells in trypsin for a very long time, and when you do not wash it, as it was shown in the video, what will happen is basically there will be enough trypsin in the vessel, and the cells would not adhere to it, right? So so when there is over-trypsinization, this is called as over-trypsinization. When there is over-trypsinization, the cells will not adhere to it. That is something that happens.
Okay. Neha, you can go ahead and ask your question.
Good morning, sir. My question
Hello,
is,
hi. Yeah.
can we use these organ on chips for understanding immunological responses in the case of transplant rejection?
Very much, very much. As every one of us would agree to it, that the scope of organ on a chip is humongous. I mean, we can study different, like, for example, if I have to talk about organ on a chip, people are also coming up with making organ systems, like incorporating different kinds of, they will have one particular organ on a chip, second particular organ on a chip, and they will connect it. So the scope is humongous, and yes, there are studies. Again, this doesn't lie in the field of my study, but I know it for a fact that yes, people are also trying to study. People do study immunological reactions quite extensively. So in that case, the cell types that they use would be different.
Thank you, sir.
No problem. No problem.
Okay, the next question again, Yugang has another question, so I'm just going to read it out for him. Okay, here's another question. So how does biofilm thickness and growth, how is it regulated to prevent overgrowth? And what are the limitations of the organ on chip? Can it be reduced by increasing the scale?
Okay. Hi, thank you so much for the question. Now, the whole agenda behind doing the experiment that we're doing is not to regulate thickness. I mean, I'm sure we can regulate the thickness by incorporating certain using less inoculum volume. The initial seeding density is what I call, or imaging at earlier time point. But that is not the main motive. We want to study biofilms as it is in native condition. So we don't regulate the thickness or the growth as such. But yes, you can do it, by imaging early on, or you can add little less density than what I use. So it really is dependent on the experimenter as such. And about the limitation of organ on chip, see, there are certainly limitations when you talk about organ on a chip. Can it be reduced by increasing? No, I didn't get this question. Can it be reduced by increasing the scale? What does that mean? Are you talking about the scalability of organ on a chip?
Yeah, the scalability of it basically.
Okay,
That
okay,
perhaps right now we are deploying it for elementary purposes because it's novel. So perhaps down the
No,
road we can.
I would disagree to it. We are not using it for elementary purposes. Organ on chip is not used for elementary purposes. We are certainly extrapolating a lot of data from organ on chip models. For example, I'll give you a very small example of what kind of experiment. Since we work on wound infection on chip, I'm trying to propagate that. I'm trying to spread that. Now, for example, if we want to study, say, wounds. Now, I can also from the reservoir was where the IVWM was, the clinical fluid was, and the outlet is where, spent medium is collected. Now, from this, I can, sort of take the medium out. I can do an ELISA to find out what proteins are there. I can also do, sequencing and to find out what is upregulated and downregulated. We have a long way to go in organ on chip systems, but then I would not say that we are trying to do elementary experiments. The experiments that are happening are very much advanced, and you'll get to know as and when you see more and more organ on a chip like systems.
Okay. I think Sachin has one more question. Can we create scratch using a pipette on this device?
Yes, we can do that. So I have done that because now the device that we use is basically a palm-sized device. So I do that. I do it using a 200 ml sterile 200 ml micro tip. So I would make a physical wound, a scratch to it using that, and we could study that. We could study migration and proliferation over the period of time. So yes, this system is very much compatible for that.
Okay, one more question from Yugang. You can go ahead, Yugang.
Yeah, so just one more question, which was regarding Milo, that what is the concentration or composition, and does it differ for different organs or different experiments, or does it remain the same?
Yes. Okay. So primarily how we have tried to develop Milo is, and normally any researcher would do an experiment is, they would try to do a thorough literature review, which is what, like people before me, Snehal, who's right now moved on, and Karishma, they had done a thorough literature review, reading a lot of clinical journals, and they came down to the components that we use. Now, the components are your FPS as a base medium. We have lactoferrin, lactic acid. We have fibronectin, fibrinogen. All of these are basically they are matrix components. They're biochemical factors. Something that is observed during an infected state. So mostly, and people do use different versions of this milieu. Depending on what their study is, basically their field of studies. But in our case, we did a thorough literature review, and we shortlisted these components using clinical journals. Okay, yeah, that's a very good question.
I'm just gonna
I really required it.
invite Shreya on to answer this question. So maybe Shreya, you can go first. Kasturi, do you want to come up and ask your question?
Yeah, sure. So I'm absolutely delighted to see your work, and both of you have been forerunners in this massive experiments. Setting up the stage has been probably not an easy task. So I wanted to ask you both, how did you deal with this absolutely new field after your master's, where you don't necessarily learn such things, and did you acquire new skills?
I would definitely agree that I did learn on the job. So it has been a good experience. So bit difficult initially, but it got easy, and then depends on your interest.
So in all disclosure, I am a microbiologist by degree. I have done my master's and bachelor's in microbiology, and animal tissue culture, to be very honest, was very new to me. I know my senior Radhika, she taught me, my SRF, she taught me, and I remember the first time she wrote my name on a T25 flask, I was so happy. I mean, I knew it for a fact that this is going to be my flask. It's not good. It's not an easy peasy task, of course, but it's always been, I mean, of course, the supportive environment always helps. Karishma has been a very great mentor to both of us, and she's always given us a stage to do whatever we wanted to do. That's important. But apart from that, I think a lot of reading, a lot of doing experiments, cool experiments, a lot of thinking is what has driven me. This field was very much new to me as well as Shreya, because Shreya is a bioinformatician. She is not, didn't know about biofilms back then. But she's learned great things. So yes, it is a bit challenging after masters, but once you're into that field, once you are interested and curious to know your own results, and when you're very happy about your positive results and want to celebrate it, it all adds up and it all makes sense. So this is what I used to think when I joined, and I thought this is going to be an impossible task for me. I mean, to assemble everything like peristaltic pump and macrofluidic device. And I know I've had like many sad days when things did not work out. There would be leakage and contaminations, something that happens, and all of us can relate to, Anushka, and Kasuri. But it's been endearing, and I loved every bit of it. I love every bit of what I do. Yeah.
Thanks.
Okay. I think there's one more question. Khushi, you can come up and ask your question.
Sir, do biofilm height
Hello.
help in determining the antimicrobial concentration?
No, I would not. Okay, so if you talk about concentration as such, I would say it would help to determine, it would help to determine the effect of an antimicrobial agent, but then you cannot extrapolate. For example, I'm sure you have heard of MIC, these minimum inhibitory concentration experiments. Now your growth is not going to ascertain how much concentration of antibiotic. You would also always know that whether this concentration is helping or not. So in our case, what we do is we measure mean fluorescence intensity and height to see whether a certain antibiotic is effective. So what I will do is, me and Shreya normally we will do is, we will vary the concentration of antibiotics and check how much reduction in height is observed. And this would be like we would know that now a certain antibiotic is effective or not.
Okay, sir. And the biofilm height also would depend on the environment, like the microfluidic
yes.
environment.
Yes, absolutely, very much. So this is an observation that we have seen. Now, normally what people use is Luria Bertini broth, right? And what in our clinical wound milieu, right? The MIC levels are different because there is an added factor of host components to it. So it's not as simple as growing in Luria Bertini broth and ascertaining antimicrobial. It's always good to go for a more human relevant system and check the resistance level or inhibitory concentrations. That would be a good measure to go forward, actually. Also, please call me Nizam, not sir.
Thank you,
Yeah.
sir.
Yeah, Nizam. A pretty nice name. Please call me Nizam.
Okay, I don't think there are any questions left. So I just want you, Shreya and Nizam, to maybe wrap up with a few closing words if you have anything to say.
Shreya, you can go ahead and I will follow you. You can.
I really believe like you could check out our work. Like you got a glimpse of it, but maybe there are papers available. You could visit our lab website, and you could find like more information about it. Nizam?
Hello, hi. First of all, I really want to congratulate the CPHMS team because I know the conditions under which they have worked, and the outcome looks so good. It's amazing. It's so when you do your experiments, I mean, it's cool and everything. You celebrate your positive results, but when some people are beautifully able to capture the essence of what we do and make us scientific celebrities. It really is very much enduring, and like I was very happy to see the whole result. So congratulations to the team. I mean, it's well deserved, I would say. I mean, you can check out our work. There is a lot of work happening in organ on a chip system, and I am sure these people would show you more colors of organ on a chip. They will incorporate more people who are working in the field or other field. But there's a lot of exciting things happening. It's not elemental anymore. Some some people are actually considering replacing them to animal models. We are not there yet. But I hope we get there sometime. And thank you so much for doing this, actually. Thank you so much. Yeah,
Okay,
Nancy
this
has a very important question. Okay. So you want to know whether there are any competitive exams after masters. So I will tell you how yes, there are competitive exams. You can give your CSIR net, you can give DBT to get into a research program. And you do get paid, because a lot of times people are asking me as a researcher whether you get paid or not, because they consider me as a student, which I am not. So you can give these entrance exams like GATE, CSIR net, DBT, ICMR, so on and so forth. Now, and you can get into a research program. Myself and Shreya, we are not PhD students per se. We are researchers working with Dr. Karishma. We just cracked an interview. I mean, the usual process, just emailing and your resume, cover letter, cracking an interview, stuff like that. So yes, you can give competitive exams after masters.
Wow, that's amazing.
Yeah, thank you so much.
I would like to add, like, it doesn't just have to be after masters. You could do while you're studying as well. Just try sending some cold emails, and maybe somebody would reach out.
I think you need to explain them about what cold emailing is. It's so important. Cold emailing is so important. Just read anything that interests you, any field that interests you. Look up scientists that are working in that particular field. Read about their work. I mean, people have fantastic lab websites. We have, like, you can check out our website. It's www.karishma Kaushik lab I'll post it. I don't remember it, but I will do that. So you can read up, and you can always email them. You can or you can always email them. You can tell them how interesting their work is, and I mean, you can also like I know for a fact that people also put up proposals that we intend to do this, we intend to study this, and this properly fits in your lab. Thank you so much, Anushka. So you properly do that, and you can email them, and that's how cold emailing works. Randomly, not like you need to know about the lab, of course, and you can send your CV, your covering letter maybe, and explaining how you're very much interested and well suited for this lab. It should be both ways. Even they will benefit out of you, actually. So always do this. People are on look for researchers all the time.
Yeah, thank you.
Just to add to that, like, for example, you can also like get a taste of different kinds of research by doing summer internships. Like, many organizations provide that. For example, Indian Academy of Sciences, and there is also JNCSR summer fellowship. So there are several. I'm sure there are more which have come up
Khurana
now.
scholarship also, which has opened up now.
Yeah, so either you can apply via these summer fellowships, like academic fellowships, or you can write to these researchers for doing like a 2-month summer fellowships directly. So yeah, just like Nizam said, right, just write to them and you'll be surprised how many people actually reply back. If you're interested, so you can do like a short stint, like a 2-month thing, just to get a taste of what these experiments are. And yeah, and many of them would be very happy to kind of take you.
Okay, thank you, Nizam and Shreya, for your time, for being with us today. We hope that, I'm just going to get to questions about the assignment. So, we hope that you enjoyed the video, and please send us your feedback. There's a link in the chat box. Give us your feedback. Tell us how we can improve. Tell us what you want to see. And so, talking about the next video and the assignments, you all must have received an email which has the B2F Circle workbook in it. If you have not received it, please send us an email and I will get back to you on that. So there are a few assignment questions for you to answer in that. The assignment is open now, and the link will be found, if you go to our link tree, you can find the submission page over there. So you can upload your assignments. The deadline is February 24th, because on February 25th, we will be hosting the P2F circle with Dr. Karishma Kaushik. She's going to come and answer all of your questions. There's going to be a fun discussion with her. We're going to do a quiz and take a look at some of your assignment answers. So you can go ahead and get started on that. In the meantime, we will get started on working on the next video for you guys. And, we hope you had a good time, and we're looking forward to hearing your feedback. So thank you everyone for joining us this morning.